Tonight, we're so fortunate to have our friend and colleague, Dr. Robert Bertin, with us. And Robert is someone who is widely admired, respected, and really loved by the botanical community in the region. So it's a real treat to have him here this evening. Let me tell you just a little bit about Robert's background. He earned his PhD in biology from the University of Illinois. He taught for 2 years each at Miami University and Bucknell University, followed by 36 years at College of the Holy Cross in Worcester. And now he's an emeritus professor of biology. His teaching responsibilities included courses in ecology, environmental biology, and field botany. His recent research has involved studies of the flora of southern New England and how it has changed over time. He's the co-author of monographs on the flora of Worcester County and the flora of Franklin County, Massachusetts. And I just want to give an appreciation for Robert as a member of our society. Not only is he our dear friend and colleague, but he contributes significantly to the success of this society. He's served on numerous different committees and in different positions over the years, and he's also had a number of special publications, including some of the floristic work that was referenced in his introduction. And I also want to just point out a special memory of him being the inaugural recipient of the first, Merit Linden Fernald Award back in 1992. So our august Dr. Robert Bertin will talk tonight on two tales of floristic change in southern New England, orchids and northern species. Welcome, Robert.
Thank you very much, Melissa, for that very generous introduction. As Melissa stated, I've been studying the flora and changes in the flora of southern New England for the last 2 or 3 decades. And I'm going to share with you a couple of stories that come out of that work. And I should, first of all, point out that there's my introductory slide. I should point out that both of these projects that I'm going to talk about were done with collaborators, and there's certainly folks here that are familiar to you. The first part of the talk on orchids is being done with Glenn Motzkin, formerly of Harvard Forest, and now a very, I think, satisfied freelance botanist. And of course, Karen Searcy, who was for many years curator of the herbarium at UMass Amherst and is now enjoying retirement in the San Diego area. My coworker on the Northern Species Project was Caitlin Spind, a Holy Cross student who has since gone on to do graduate work in marine ecology. So the two parts, I'm going to first talk about orchids and then about northern species. In the two parts of the talk, there'll be kind of 3 things that I address in each or touch on. One is, have these plants changed over time? That is, have the distributions of those plants changed over time? Secondly, if they have, what have been the causes? And then thirdly, which kind of runs through a lot of this, I want to talk about the real challenges one has in trying to determine whether changes have in fact occurred. And this is both lies in the difficulty of getting appropriate data to use, and also difficulties in interpretation of the information that we do have. So those 3 themes will be present in both parts of the talk. So let's start off then with orchids. I guess you probably have all heard of the term charismatic megafauna to refer to peregrine falcons and wildebeest and so forth. And I don't know if botanists actually have a name for the equivalent, but I would say orchids are it, if there is such a thing. This is a very diverse family. Depending on who's doing the counting, it's either the most species-rich or the second most species-rich family of vascular plants. Many of them have spectacular flowers, of which these are a few examples, although I hasten to point out that that's not true for all of them. Many of them have very non-spectacular autogamous flowers. Okay, so we have the whole gamut there. We certainly have many with unusual pollination mechanisms. There are your conventional insect pollinated species. There are ones that produce no nectar and appear to rely on mimicry of something else to get to fool the pollinators. And then there are a number of them, including this one over here, that mimic female insects and are pollinated in a mechanism called pseudocopulation, where the male thinks he's found a receptive female, attempts to mate with it, picks up pollinia, and then is confused again and transfers the pollinia to another plant of the same species. There are also, as an alternative to conventional sugar-based nectars, there are many orchids that produce oils that are collected by euglossine bees and are used in their own mating rituals. The seeds of these species are dust-like. They're very, very small. Most people say they have no endosperm in them. And so they're numerous, very small. And all species, as far as we know, have an obligate dependence on fungi in the early stages of their existence. And this is relating to the fact that the seeds aren't carrying much food. And orchids appear in most cases to live underground for a few years, dependent on fungi, before they first emerge above ground. And there are certainly some species of orchids that have an obligate dependence on fungi through their entire lives. So a lot of interesting biology represented here. Most of the diversity of orchids, of course, is tropical and mostly epiphytic, but we have a subset of that, which is still reasonably large for our flora, I should say. Approximately 51 orchid species can be found in the southern New England states, Massachusetts, Connecticut, Rhode Island. And I say circa because their taxonomists are busy at work dividing species and doing the things that taxonomists do so well. And for example, this number 51 does not include the recent segregates of Spiranthes cernua, which is now Archaecephala, and a few other things. So that's why I put that as approximate. And it turns out Franklin and Worcester County, Massachusetts will have a special role to play here. And in fact, 44 of the 51 species you can find in southern New England, you can find in those two counties. So what kind, if I want to get at the question of whether orchids are declining, which has been suggested, what kinds of data do we have available that we might use? These would seem to be the four main possibilities. Herbarium specimens, of course. And we have a record in this part of the country going back to close to 1800 for the earliest herbarium specimens and continuing up to the present. Secondly, since 2008, we have had this digital platform, iNaturalist, where people take photographs that are georeferenced and put them on the site. And there's a database now of millions of records of different plant and animal species that you can find on iNaturalist. So that is a second possibility. Those records, as I say, date to 2008 when iNaturalist was begun. Thirdly, we have records from the natural heritage programs in the different New England states. All three southern New England states have natural heritage programs. They track information not for all orchid species, but for state-listed species. And they're pretty thorough about this. They have gone around to herbaria to try to find the historical records, and they keep track of every known population of state-listed species in their state, and they try to revisit them every 25 years or less. So we have pretty good data on those listed species. And the map I show you here is from the Mass Data Viewer. This is on the website for the Mass Natural Heritage Program. And this particular one is for Platanthera dilatata, and indicated in those red, the red towns are the ones in which that species has been collected at some time. Then finally, we have some detailed floras that have been published for 2 New England counties with town level distribution data. And the 2 counties are Worcester and Franklin County, Massachusetts, which is the reason that I singled those 2 out. And both of these floras draw on herbarium records, but they also include a lot of de novo field work. And so they have pretty good documentation of what is present in those counties today. All right, so these are the data sources we have available. How can we use them? Well, first of all, of these 4, the historical data really have to come from herbarium specimens, because that's really the only good record that we have of those. And you might wonder then, could we simply use the number of herbarium collections as an indicator of abundance of the species? And I think you're all savvy enough to know that unfortunately we can't do that. And I'll illustrate that with a non-orchid species. This is probably the most common woodland herbaceous plant in Massachusetts, or right up there, Myanthemum canadense. And in fact, the 553 records are from the CNH digital repository of New England specimens. And if you just take those at face value, you would say, well, this species increased from a low level in the 1800s to a peak at around 1900, and then it's been gradually declining since. And of course, there was a big drop during World War II. And I think you all realize that that is totally false. What we're really looking at here is collection intensity, okay? And so we can't, unfortunately, simply take the numbers of herbarium collections as a record of abundance. It just doesn't work. But they are very useful. Notice this period here that you might say is sort of the golden age of collecting from the late 1800s through the early 1900s. There really were very widespread collections at that time. And so there it strikes us that that is a pretty good record of the historical frequency and distribution of most species. Well, how about if we take the herbarium specimens as a historical record, then how about the current record? Can we use iNaturalist records, for example, as a good indication of current frequency and distribution? To get at this question, we used the 2 counties for which we have detailed floristic information. Those are Franklin and Worcester County, Massachusetts. And we took 10 orchid species that are not state listed species, because a lot of the iNaturalist records of state listed species are suppressed. Okay, the location is suppressed for obvious reasons. So we took 10 non-listed species and looked at how good iNaturalist was at capturing the number of towns in which they are actually known. Okay, so these are our 10 species. This column here represents the number of towns in which those species were found in those 2 published floras for Franklin and Worcester County. Worcester County has 60 towns, Franklin County has 26 for a total of 86. So you can see that those floras recorded pink lady slipper from 85 of the 86 towns, okay? And varying numbers. Calipogon was only found in 6 of the towns and so forth. This column gives the percentage of those towns for which iNaturalist has records. And these would be 2008 or more recently. And you'll notice 2 things about that. First of all, iNaturalist records capture less than half of the towns in which the species are known. And I might say that these numbers over here have to be minimum numbers, because we couldn't be everywhere and see everything, okay? So somewhat less than half of the towns in which a species is known were captured by iNaturalist, but perhaps more importantly, notice the variation in these numbers. iNaturalist does a pretty darn good job of capturing pink lady slipper, and that's not perhaps too surprising. It's a relatively common species. It's showy, it's easy to identify. It tends to grow along trails that people walk on in the upland and so forth. But then take a species like Corallarhiza trifida, pale coral root, which is an inconspicuous species that grows in damp areas, and you have to be looking closely to find it. It does a poor job of capturing that. And that variation in those numbers is a problem, because that would give us very biased results if we simply relied on iNaturalist data. Now, I also threw this column up, the percent of towns with recent herbarium specimens, and notice that that does even worse than the iNaturalist data. Okay, so we clearly can't use recent herbarium records as an indication of abundance. All right, so given all these caveats, what does this leave us with as far as what we can actually use? We came up with the following. For state-listed species only, we decided that we could use historical herbarium specimens and recent natural heritage records, because natural heritage does a very thorough job of keeping track of those populations. And we defined a historical and a recent period. The recent period, it covers a period of roughly 25 years and a year or two for reporting, because they have a 25-year look-back period, okay? And so anything from 96 on was considered recent, anything 95 or before was considered historical. Notice, however, this applies only to state-listed species and only in those states in which they are listed. That might be just Rhode Island for some of these orchids. It might be Rhode Island, Massachusetts, and Connecticut for others, okay? But those are the limitations of the data. Now, for other species that aren't state listed, we felt we could not make any use of data that covered all of southern New England, because we just don't have the data. There, we felt we were limited to Franklin and Worcester County, both of which have detailed recent town floras with town level distributions. And in each case, we defined a recent period that was depending on when the floristic work started compared to the previous period. The Worcester County floristic work was done slightly earlier, so this is the recent period for that and the historical period for that. All right, so a little messy already. We've got data for state-listed species in those states in which they're listed, and we also have records from 2 counties. Now, we wanted to define a measure that represented change. We didn't want to simply rely on disappearance from a county versus still present in a county, okay? So we came up with a change index, which is defined as follows. We took the number of recent towns minus the number of historical towns and divided it by the sum of those two. So for example, if a species was found in 3 recent and 6 historical towns, it's 3 minus 6 over 3 plus 6, minus 3 over 9, minus 0.33. Okay, thing to remember here, the range of change index, the possible range is -1 for something that's disappeared to +1 if it has appeared, and it's 0 if it's found in the same number of towns in the historical period and in the recent period. Okay, so fairly easy to understand there. All right, well, first thing we did, we wanted to know whether the information that we got from Franklin and Worcester County spoke at all to what was going on in southern New England more broadly. So the first thing we did is to look at those species for which we had both southern New England data and also Franklin and Worcester County data. This would be state-listed species that happen to occur in those 2 counties. Okay, here we calculated a change index for all of the orchid species that met those criteria. So each point here is 1 orchid species. This is the change index for southern New England. This is the change index for Franklin and Worcester County. And you'll see that, although perhaps not as tight as we would like, there is a strong positive relationship between these 2 variables. The correlation coefficient was 0.53. The significance of that was 0.001. And I might point out that a number of the state-listed species were only listed in Rhode Island. Well, Rhode Island has much to its credit, but it is a very small state, and therefore it gives you a pretty small sample of towns. particularly considering the orchids aren't present in all of the towns. If we were to exclude from this graph species that were listed only in Rhode Island, so they had to be listed in Massachusetts or Connecticut and possibly other states, then the relationship actually gets quite a bit stronger. We get a correlation coefficient of 0.72 and a significant, more highly significant relationship here. So this suggests to us that although there are going to be differences here, there is a strong and significant relationship between what's going on in these two counties and in southern New England more broadly. Okay, that was the information I just gave you there. All right, so results, what do we get? First of all, in this graph, this part of the graph should sit underneath this part, okay? They're not two separate graphs, but one very tall graph that I couldn't fit on here. This is the zero line for the change index here and over here. And you'll notice it's pretty strongly tilted on the left side there. Okay, there are only 2 species that have positive change indexes out of all of those orchid species that we looked at. And some of them are -1. Okay, in fact, the top 6 species are -1. They're historical in the towns, in the states where they were listed. And because there are so few positive, maybe we should look at those. Okay, the one right at the bottom is Neotia bifolia. That has a change index of +1. That's because it was not known in southern New England until, I wish Bob Werner were here, he could tell me, either 2008 or 2011, something like that. It was discovered. And I think it's known now in 1 or 2 towns in Massachusetts. Now, did it appear by itself? Was it always there but overlooked? My guess, probably the latter. Okay, I can't say for sure, but my guess, and you can see that it's not an especially conspicuous species. Okay, so that accounts for that species. And the other one there is actually another inconspicuous species, Coralrhiza odontorrhiza. Okay, and notice I would hesitate to argue that that is significantly different from 0, but at least it's on the positive side of the line there. Every other orchid species is on the negative side, and most of them very substantially so. Now, you might say, well, these are state listed species. Of course, these are going to be species in trouble. You know, maybe I'm biasing my, so let's look at Franklin and Worcester County, where we have a more complete list, okay? Okay, same graph setup here. And notice again, there's a preponderance of species on the left side of that 0 line. In fact, this is significantly different from what you get by chance. And I might point out one other thing, and that is when we did our sampling in both of those counties, we were very systematic about trying to accumulate town records. And as a result, if we took the change index for all native species, the average change index comes out to somewhere between 0.1 and 0.2, plus 0.1, 0.2. Now, we don't interpret that as meaning that native species are spreading to new towns. Rather, we assume that is a function of the more intensive and systematic sampling in our floras. So really, in a sense, the 0 line probably should be at about 0.15 or 0.18. And in fact, if we put it there, which would be right about here, then there's maybe 4 species that are positive and a couple of those barely so. Notice, by the way, that the bottom species, the one that's increased the most, is our one non-native orchid, Epipactis helberine, which is increasing. Interestingly, notice the second one from the bottom is Coralrhiza odontorrhiza, which is the same species that was nudging on the positive side in those states where it is listed. And notice also that the other ones down here, most of them are common species, the Spiranthes cernua complex, Cypripedium acaulis, Goodyeara pubescens. And so I think the fact that these are positive is just the fact that they're probably present in every town, and we were just a bit better at systematically sampling the towns. All right, so pattern here seems to be very clearly decline. Well, This is a graph where I pulled out data from other comparative studies in the Northeast. Most of them are Massachusetts, but there's one from Pennsylvania and New Hampshire and Maine and New York. And in each case, I looked at, in this case, losses. I couldn't calculate a change index because they didn't get their data in that way. But here they're looking at losses of orchids. And notice that in 13 of 13 cases, a higher percentage of orchid species are lost than of all species in the flora. Okay, the likelihood of that happening by chance is vanishingly small. Let's look at a couple, some individual, one other thing. A methodological note. One of the points that has been raised with orchids is that perhaps what appears to be loss is actually just a function of the fact that most orchid species are relatively rare. And if you compare orchids to the whole flora, you're comparing a group of largely rare species to a group of species, many of which are very common. So maybe that accounts for the apparent loss. Well, the only way to get around that is to compare orchids to other species that have a comparable level of rarity. Okay, we have that information for Franklin and for Worcester counties. And so we did that in this next slide, where we grouped both orchids and non-orchids according to the number of towns in which they were known to occur historically. And then we compared the change index of all species versus orchid species in each of these categories. And notice that except for this last category here, all of the orchid ones are more negative than the other species. And I think there's reasons for this over here. These are the common species. It's pink lady slipper and Goodyera and so forth. And those are probably present in all of the towns and remain present in all of the towns. So difference here, highly significant. Okay, so this is the first analysis I know of where orchid decline, where basically we've controlled for rarity in making our comparison, which I think is important. It's always nice to have some maps and some look at particular species. So Calopogon tuberosus, all the maps will have the same legend here. If it's black, there are both historical and recent records, dark gray, recent only, pale gray, historical only. So in a case like this, you see a lot of pale gray, okay? And this is a species which is apparently declining. It has a negative change index. Long bracted orchid, this is had one of the most negative change indexes of all of the species. We found it in 2 towns in those 2 counties, and there's a whole bunch of towns in which it was known historically, not relocated. Showy orchid, still doing pretty well. You can find it in a lot of places in Franklin County. I don't know of it anywhere in Worcester County, and it's also dropped out of some of the Franklin County towns. Platanthera flava, which is a state threatened species. Again, lots of light gray indicating towns in which it apparently no longer occurs. Slender ladies' tresses, Spiranthes lacera. This actually had one of the most negative change indexes of all species, and I actually suggested to Bob Wernerl that he might want to consider putting this on the state watch list or state listed. He pointed out to me that it was actually seemed to be doing pretty well in the Cape and Islands, okay, that there are some good populations of it there. So that's not something that we picked up here in Franklin and Worcester County. And then for something quite different, Epipactis helberine, the introduced one. Okay, so that's in a lot of places. And my guess is it's present in every town. But okay, so move on. What is causing these declines? And a lot of variables have been suggested. Some people have suggested it's an artifact of sampling. One of the things that we can't really control for here is our recent samples are based on a time period of like somewhere between 10 and 25 years. The historical samples are based on a period of 150 or so years. I think our sampling was more intensive. So we're kind of comparing a shorter period of more intensive sampling with a longer period of less intensive sampling. But I can think of some reasons why you might pick up orchids over a longer period where you might not find them over a shorter period. And that's just something we have to live with. I mean, not all orchid species come above ground every year, for example. And also, you figure if it's a longer period of sampling, that probably has seen the landscape in more different successional stages than we see it in a shorter period of 20 years. Anyway, so there are sampling issues that we can't get around. Succession, herbivory, possibly deer, possibly slugs, introduce slugs and other things. Introduced species, not so much plants, but earthworms, eating that organic matter on which those fungi depend. Soil acidification, soil nutrient enrichment from nitrogen deposition, climate change. All of these things have been suggested. I'm going to report on a subset of these. This is the work that I'm doing with Glenn and Karen, and here we looked at 4 variables: habitat, leaf size, which we thought might be related to herbivore interest, pollinators, and location within the plant range. Are these northern species, southern species, and so forth? And I'll tell you right off the bat that our results were not very satisfactory. Let's have a look at the data. Wooded versus open habitats. Clearly, no difference for the listed species in southern New England. In Franklin and Worcester counties, the species of open areas weren't doing as well, but notice this is not significant, the difference here. But this would certainly be consistent with the afforestation that we have seen over the last century. And I would be willing to bet that there are certain species like slender ladies' tresses that are probably where this may be the most important variable. But overall, I don't have a significant result to show you. Leaf area. Here's a plot of leaf area. We basically modeled them as ellipses. And this is change index, did a regression of change index on leaf area. We have a regression with a negative slope, and the p-value is tantalizingly close to 0.05, but not quite significant. This would be an example of a large-leafed orchid, Platanthera orbiculata, and Trifera is a small-leafed one here. Interestingly, that species right there is the one that led off the talk in the Cypripedium regini, the showy lady slipper. That has the largest leaf area of all of our orchids. And there's a very interesting study that was done in West Virginia a number of years ago, where there were a couple of showy lady slipper populations that deer got into and browsed intensively for a couple of years, and then they were fenced and protected. Even after 10 years, not all of the variables of those populations had returned to their pre-browse state. Okay, so it was a very clear indication of the level of threat that herbivores can impose, but not quite significant. Okay, insect pollination. That is, this compares autogamous species and insect pollinated species. Not much difference here. Here, there was a difference. The insect pollinated species have more negative change index than the autogamous species. And this difference, again, is tantalizingly close to significant, but not quite there. Okay, so maybe there's something going on, maybe there's not. If we look at the different insect pollination groups, the bee-pollinated ones in general were doing better than the lep-pollinated ones, which were doing better than the fly-pollinated ones. But the difference was not significant. A lot of variability here. Okay, the last thing that we looked at is, do northern species and southern species behave differently? And so here for every dot is an orchid species, and we gave a latitudinal index to each species. These are the northern ones here. These are the southern ones here, a couple of examples. And when we regress the change index on the latitudinal index, my goodness, we actually have a significant result here. And notice that it's the northern species that are not doing as well as the southern species. Well, this is the pattern that you would expect to see if climate change is having an effect on the distribution of these things. So that's the one significant result that we got out of all of these analyses. And we did a few more in addition, and I won't bore you with the details of those. They weren't significant, though. So let's see, I believe that actually wraps up that portion of the talk. Yes, that wraps that up. So I guess we'll deal with the questions at the end, but I'll be happy to take those then. Let's move on then and talk about northern species, and this part won't be quite as long as the other part. And Linnea borealis here would be a poster child for a northern species in Massachusetts. Now, why would we look at northern species? Well, if this is the range of a northern species, we call them northern because they reach their southern limits close to New England, southern New England here. And if we had a certain amount of warming that pushed this range, the suitable climates northward, then we would expect them to drop out of the southernmost towns in their range. On the other hand, if there was a species for which Massachusetts was closer to the center of its range and we have a comparable climatic change, we wouldn't necessarily expect to see any difference. So we were interested then in identifying a group of northern species for which we had good historical and current data. And we figured the best ones were the rare species because natural heritage is keeping track of those. We figure we have pretty good herbarium records for most species, but it's the rare species that we have the best modern records for southern New England as a whole. So we went through the list of state-listed species in Massachusetts and extracted a group of northern species. I won't talk much about it, but we also extracted a group of southern species and we subtracted a group of species that was neither northern nor southern for comparative purposes. Okay, but I'm going to set that part aside. These are some examples of species that are on that list of, I think it was about 26 species: Carex pauciflora, Sorbus decora, there's twin flower again, and Ribes lacustri are just some of the examples of northern species. So what do we find about their change indexes? Oops, methods first. We did the following. For each species, we generated a list of towns where they were known historically and towns where they were known in the recent period of about 25 years. We then calculated an average temperature. This is a 30-year average temperature for each of the towns in southern New England. There are some climate data from this PRISM group out at Oregon State that were useful for that purpose. So we now have an average temperature for each of the towns in southern Massachusetts. And as you would guess, that's influenced by latitude, and it's also influenced by altitude of the town. Okay, those are primary variables, probably also somewhat distance from the coast, but in any event. So we now have, every town has its average temperature. And then we took, for a given species, we took the group of historical towns and calculated a temperature average, and the group of recent towns and calculated a temperature average. If they're dropping out of the southern towns in their range, that means the recent towns should have a colder average temperature, right? They're moving north. They're not moving north, rather they're getting wiped out of the southernmost towns. All right, so what did we find? This represents zero temperature change. Everything to the left side of that is a species in which recent towns have a colder average temperature than the historical towns. Everything to the right of that line, and you can see there's really only three that are consequentially to the right, those represent species in which the current towns are warmer than the historical towns. An overall analysis on this group of species shows there's a significant tendency for them to be present in cooler towns today than they were in the past. Now, if life were simple, We could end the talk there. But one of the problems here is that this type of analysis of necessity is a correlational analysis. So one of the possibilities might be that there's some variable that's correlated with town temperature that is actually a causative variable. And it has nothing to do with temperature at all. And one of the ones that occur to us, I don't know, maybe can you think of any that might be correlated there? In southern New England, some consequential variable for plants. How about the human imprint? Would you say that's more in the south and lower elevations or the north and higher elevations? I'd say south and lower, okay? Well, obviously, human modification of the landscape can have immense effects on plants and all kinds of other things. So we thought we'd better look at that. And so in addition to getting an average temperature for each of the towns in southern New England, we also calculated an human population density in each town. I think we used 2010 census data. Is there a relationship? Oops, sorry. I've jumped ahead of myself. Okay. Put that thought on hold for a moment. This just gets into the details of some of those species that I presented on the previous slide. It so happens that these are the 6 species with the largest sample sizes and therefore presumably least subject to sampling error. This is the number of total towns. This is the number of towns in which they're found historically. This column is the number of towns in which they're found currently. Notice, first of all, that in every one of the 6 cases, they're found in fewer towns today than historically. And then this is the average temperature of the historical towns and the average temperature of the recent towns. Notice that in each of the 6 cases, recent towns have a lower average temperature. Okay. And the likelihood of, just for these 6 species, of that occurring by chance would be 1/2⁶, or 1 in 64 chance, or something like that. So less than 0.05. Data on a couple of particular species. Linnea borealis used to be found widely in southern New England. It's now gone from the Cape, gone from Rhode Island, gone from Eastern Mass. As far as we know, it's hanging on in a couple of towns here, a town up here, and every other town is in the northern Berkshire Plateau or the northern Worcester Plateau. Okay, this had a temperature change in those towns of about 2 degrees centigrade or something like that. Another one, Carex lenticularis, there used to be populations here, one down here, supposedly. And now the only place they're found is on the upper Deerfield River. Okay, and when we were doing our Franklin County study, we actually looked through all of these towns and we didn't see it. It may be there, but we didn't find it. So this is a couple of examples of species that have undergone those changes. All right, so back to the point that I was starting to make about population. Maybe this could be just due to human population density. We did a correlation between average temperature of a town and the average population density in the town. And notice there is a highly significant positive relationship between the 2 variables. Well, that's a bit of a conundrum. We have 2 correlated variables that might account for the changes that we've seen in these species. And fortunately, this is a case where statistics rides to the rescue. In effect, what we did is we performed the regression that you see here and then calculated the residual or the deviation from the regression line. That represents variation in the town temperature that is not explained by human population density. Okay, because we've used this regression to explain the variation we can from human population density. Then we took the residuals, and we would expect to see more negative residuals if current towns in which a species is found have lower temperatures. And lo and behold, the relationship we found originally still holds. Okay, it doesn't look highly significant here, but in fact it is. Okay, so these represent the historical towns with their higher residual. These represent the recent towns with a more negative residual. Okay, so the point from this then is we've corrected our values for human population density in each town, and the relationship with town temperature persists after that correction. All right, so conclusions. Orchids. Frequencies of almost all orchid species in southern New England are declining. The causes, and I've emphasized the word may, include herbivory. Climate change was one of the stronger signals, pollinator decline, land use changes. Our suspicion is that there are multiple variables involved here, and the variables that are most critical differ from species to species. Certain large-leafed orchids, it may be herbivory that's most important, and a species like Spiranthes gracilis or lacera, it may be something like the afforestation of the habitat. As far as northern species go, rare northern species are disappearing from the warmer towns in southern New England, and the causes of this appear to be both climate change and human habitat change. So I would like to finish up with some acknowledgments here. Of course, my colleagues on the project, who I've recognized already. I couldn't have done this without the cooperation of the folks in the Natural Heritage Program, Bob Wernerl, Carol Frost, Sarah Meyer in Massachusetts, and Karen and David in Rhode Island and Connecticut. Herbarium assistance in several different herbaria efforts help was appreciated there. And finally, for GIS assistance, Rich Lent at Holy Cross. I might mention the northern species study has been published already, and if anyone is interested in a PDF of that, just send me send me a note, email me a note, and I'll send it along. The orchid study is currently in review at Rodora, and we hope to have that in final form within a few months. And if you're interested in that, I can send you a copy of that too. So thank you very much. And I'd be delighted to take questions. Yes.
In your study of orchid declines, you compared the rare orchid species to other rare species, which are
Yes.
not orchids. What were those species, and how did you select them?
Excellent question.
Don't forget to zoom on.
I just had an excellent question. The question was, when I compared orchid species changes to changes in non-orchid species of comparable abundance, how did I pick out that set of non-orchid species to use? We tried to match sort of the growth habit to orchids as well as we could. So we first eliminated all woody species. We eliminated all wind-pollinated species. We eliminated all species that have extensive clonal growth. There are some orchids that are a little bit clonal, like Goodyeara, for example. You see little patches, but they're not very extensive, not like fields of goldenrod and things like that. And so, and we, let's see, were there any other, I forget whether there are any other limitations, but we got a group of about 300 or 400 species that seemed a reasonable comparison group. Yeah. Richard.
Fantastic talk. This is so interesting.
Well,
so much.
Thank you.
It's really intriguing just the way you've done this analysis, because I think that what I would have thought to have done with this analysis is looking at changes in relative abundance. So you've been comparing orchids against historical, against modern town occurrences, but I mean, the way I think I would have started to do it is to see like the relative changes, to see if orchids were being observed or collected, or if orchids are being currently observed less frequently in comparison to other species, in terms of herbarium specimens or naturalist records, and approached it that way.
So, yeah, go ahead. Yeah. So,
I can think of big reasons why it wouldn't work. that's the way I would have started it. But did you think about doing that at all? Changes in relative abundance?
I'd have to think about that a little bit to see how that might work.
I mean, you could think it like, imagine that orchids would have been, say, 5% of the herbarium collections 100 years ago.
Oh.
And now maybe they're only like 3% of the observations or 3% of the herbarium collections.
Oh, that's a very interesting thought. For herbarium specimens, I'm saying I don't think it would probably work because many of us intentionally do not collect orchids today for understandable reasons. Populations are small and, this and that. I guess I'd have to give some thought as to whether we could do that with observational data. But I hadn't, to be honest, I hadn't thought about doing that at all. And I'll have to mull that around a little bit. Yeah. Lisa.
It would be interesting to run your orchid comparison with the Ophioglossales. Because the similar causes of their decline have been postulated.
Yes, yeah. The question was to perhaps compare orchid declines to declines in the Ophioglossales, which might be subject to some of the same pressures, perhaps herbivory in some cases, the same kind of mycoheterotrophic relationships and so forth. And it's a point well taken. As you probably well know, Lisa, the Ophioglossales are also one of the families that shows up as a major decliner in most of these comparative studies. Yeah. And I might point out, since you asked a question, Lisa, a little bit, I was looking through some back issues of Rhodora, and I noticed that you had published a small addendum to your Flora of Needham that had not relocated Platanthera lacera, the fringed orchid, and then you found one in your foundation plantings at your house. Is that correct? They're in full bloom. Yes. And that I think is a little cautionary note in how difficult orchids can be to predict or find. Yeah. Yeah. Yes.
From the chat I want to throw in there. So this person says, I apologize in advance if this was covered and missed, but I heard you mention average temperatures and how their increase might be correlated with the loss of northern species. Yet if you look at the USDA plant hardiness index, that is based on minimum temperatures. So I'm wondering whether it is a change in temperature extremes rather than averages that might have a greater impact on the increase or decrease of abundance in a particular location.
Oh, an excellent question. And I guess the simplest answer would be we really don't know what temperature parameters are the most important for setting the limits for most species. It could well be extreme temperatures rather than average temperatures. So that it's a point well taken, and in fact, it may be entirely correct. So I'll have to throw this out and try again. Yes.
I was also wondering, like, if you're potentially like combining the ideas of like both tails, like with certain species moving more north and like disappearing, like Cypripedium or Antinum comes to mind. I know that like they can be pretty susceptible to like higher temperatures, like in the springtime specifically. And like how like Tipularia has been slowly creeping more north to the warm climate. Were you looking into that at all?
So was the question there about?
Or were you looking at like the climate change implications with the orchids ranges?
And in particular, whether there might also be southern species moving north, perhaps. I have not looked at that specifically with the orchids, but I did look very, very specifically at that for a wider group of species when I was doing the Worcester County flora. And in that, I found that the northern species did seem to be dropping out. They seemed to be declining, number one. And number two, they seemed to be dropping out of the southernmost towns in their ranges. Southern species, however, did not seem to be increasing. And when they were found in new towns, they weren't necessarily found to the north of where they were before on average. The way And now I'm sure there are particular cases where, yes, they have moved north. But the way I interpreted those results that I found in Worcester County is that species disappearance simply means that some sort of threshold has been exceeded for the species and it's no longer able to make a go of it. And so a population can wink out fairly quickly. But species movement into an area, that is involved with dispersal mechanisms and establishment and so forth. My guess is that in many cases, that's going to be a much slower process than species disappearance. And it will be specific to the species. So something like an ant-dispersed species might have a really tough time expanding northwards. Something that is bird dispersed wouldn't have that sort of limitation. So I did not look at that specifically in orchids. Well, actually, that's not quite true. I did, because I mentioned in the orchids, was it the orchid study? No, I'm sorry. It was in the northern species study that I had a separate group of southern species, southern rare species that I looked at. In that case, we did not find them moving northward. Yeah, so I think maybe the two processes proceed at different rates. Yeah, but good question. Yes.
I thought that the data you included on population density was really interesting, but was there any thought given to, like, I guess the sheer total amount of like land that would be left available for species to grow? So instead of looking at population density, was there any consideration given towards, I guess, arable habitat would be the term I would think of?
Yeah, the question for the folks online pertained to using population density as a measure of human imprint versus perhaps using something better like some aspect of land use or something like that, or land character. I used human population density because it was convenient. And when I was doing a study on changes in Worcester County some years prior, in which I did go into the state database layers and took out all the land use layers and looked at it, I found that human population density was highly correlated with the proportion of natural habitat like woodland and wetland. And so it seemed to be a pretty good proxy that was easy to measure. Yeah, so I did not proceed past that. Yes.
So this study was done using presence data. And this is mostly like an opinion question, I guess I'm asking to you. Was the decline in presence, is that analogous to like what you were finding at the sites in terms of the quality of the populations? Or were they disconnected? Were you finding sites where the populations were still really healthy and they were declining at multiple sites?
The question pertained to my use of basically town frequency data or presence absence in towns, as opposed to perhaps some measure of the vigor of populations. And that's an excellent question. And because of the nature of the data I was using, we did not have information on the vigor of the populations, and so I could not use that. Jesse Bellmare at Smith has done some very interesting work with looking at the population level data for relatively northern species like Clintonia borealis, for example. And I think in the ideal world, we would have information at both levels because it's certainly important. And what goes on at the level I'm reporting really is a function of what's going on in the individual populations. So, but I don't have those data. Richard.
So you described the fact that orchids are declining and that northern species are losing their southern populations. So what do you advise us as a club and people in the movement to start doing? Should we go out there and start creating new populations of orchids? Should we start very actively planting new populations of these species further north?
The question pertained to what should we be doing about this decline of orchid populations and northern populations and so forth. That's a challenging question. There are certainly some who would argue that assisted migration is something that we should be looking at. No, I did, but essentially, trying out more southern species from eastern North America, and I certainly think we should be looking at that. But you know, it's kind of interesting. The assumption is that this is going to be sort of a monotonic increase in temperature over the foreseeable centuries. There are other possible scenarios. If the circulation in the Atlantic Ocean is modified in the way that it is going, we could actually be in for a much colder climate locally. And I'm not bright enough to know what the probability of that is, when it might occur, if it does occur. But there are examples in the recent geological record where a shutdown in that ocean circulation pattern caused extremely cold temperatures in Europe and in this part of North America. So what's the future going to bring? I'm not sure. I mean, the general trend for the globe is clearly warmer. There's no question about that. I'm not disputing that, but there could be local anomalies that throw all of our sort of anticipated changes out the window.